All right, now we're going to move on to some essential concepts that we really need to understand before we can move on to the body systems. So the first portion I would like to discuss is the functions of the nervous system. So when we think about the nervous system as a whole, we have sensory input. This is information that's gathered by sensory receptors about internal and external changes that are happening. So imagine that you place your hand on a hot stove. Those sensory receptors are saying, this is hot. It's going to send that information afferently towards your central nervous system. Once the information goes to your brain, it's going to have integration. So this is when our brain interprets that sensory input. So the brain is saying, oh goodness, this is hot. And what do we need to do about it? And then we have our motor output. Our motor output is going to be that efferent information that's going to go to our organs. So that's our muscles, our glands. And it's going to produce a response. So we have the efferent information that goes out to the hand and says, remove your hand from the hot stove, and then you remove your hand from the hot stove. So while we may be familiar with the nervous system, a very fundamental way to think about it is we have that sensory information coming in, it's integrated and interpreted, and then the motor information goes out and produces the reaction. When we talk about our nervous system, we have a central nervous system and a peripheral nervous system. Your central nervous system consists of two things, your brain and your spinal cord. And that is really what intraoperative monitoring is based around, the brain and the spinal cord. This is the integration and the command center. Okay? But we also have the peripheral nervous system. And the peripheral nervous system are those paired spinal and cranial nerves that carry the messages to and from the central nervous system. And we also have a huge part of intraoperative monitoring that's dedicated to the peripheral nervous system. So, our peripheral nervous system is sending those afferent and efferent information to and from the central nervous system, which is really that command center and that integration center. So now we want to specifically look at the peripheral nervous system. And as I just said, the peripheral nervous system really has two functions. It's the sensory afferent information. So this is conveying impulses from our skin, our skeletal muscles, our joints. That's our somatic afferent fibers. It also is conveying information from our visceral organs. That's our visceral afferent fibers. All of this sensory information is coming in to the central nervous system, okay? The peripheral nervous system also has our motor and our efferent division, and this is when we're transmitting the impulses away from the central nervous system to the effector organs. And again, that can be muscles, glands, different things like that. So the peripheral nervous system is sending afferent fibers, sensory information in. It also has efferent, sending the motor information out. So then we want to zoom in on the motor division specifically of the peripheral nervous system. Okay? The motor division has two parts, the somatic and the autonomic. So the somatic nervous system is voluntary. So this is when we pick up our pencil, we're taking notes right now. We are voluntarily saying, okay, skeletal muscle, I'm writing. Okay, so you're in control over that. That is different than your autonomic or your involuntary nervous system. And this is going to be where we don't have to think about it at all. Right now, what's happening inside of our body. Smooth muscle is moving. Cardiac muscle, our heart's pumping. Glands are also functioning right now. All of this is part of the autonomic nervous system. It is involuntary and it happens on its own. We don't even think about it. Okay? The autonomic system has two divisions. Okay? So 2 subdivisions of the autonomic is sympathetic and parasympathetic. So many of you may have heard of sympathetic is our fight or flight. So we are hiking in the woods, we see a bear. What happens? Our heart rate increases, that cardiac. Our palms start to get sweaty, those glands, okay? That's our sympathetic system coming into play. We didn't say, oh, there's a bear, palms get sweaty, right? That automatically happens. That's different than our parasympathetic system, which is going to be rest and digest. So this might be after we have a delicious meal and we're sitting down or lying down, and that smooth muscle and those glands start to digest that food and we feel ourselves getting tired. That's our parasympathetic function, okay? And both sympathetic and parasympathetic are part of the autonomic nervous system. So when I think about the nervous system, it really happens in this flow chart, everything that we just discussed. And this is really foundational that everyone needs to know if we are in any field of medicine at all. Okay? So when we look at the nervous system, we have the central nervous system. The central nervous system consists of what 2 things? The brain and the spinal cord. The peripheral nervous system consists of what parts or what things? Paired spinal and cranial nerves. A lot of people forget that. Our spinal and our cranial nerves are in the peripheral nervous system. So intraoperative monitoring, we're monitoring the entire nervous system, or we have the capability to. So we're looking at central nervous system, brain and spinal cord, and peripheral nervous system, spinal nerves, and cranial nerves. Okay? When we think about those nerves, all of our nerves have a sensory division, or afferent, and a motor division, efferent, right? We have sensory evoked potentials. We have motor evoked potentials. So we look at both aspects of the peripheral nervous system. Now, whenever we break down motor, we have autonomic or involuntary nervous system, and then we have a somatic or voluntary motor system. And we have different ways of monitoring both of these. Most of us are probably familiar with monitoring the somatic or voluntary, right? So quadriceps, gastrocnemius, the different muscles of the leg, those we have more voluntary control over those muscles. But we also monitor the bulbocavernous reflex. That is an autonomic. We don't control that muscle per se. So that's a way that we're able to monitor the autonomic or involuntary muscle group. And then from the autonomic, we have 2 portions of that: sympathetic, fight or flight. So we see a bear, palms get sweaty, heart rate goes up, that's sympathetic. And then parasympathetic is rest and digest. So we just ate a meal, we're relaxing, our smooth muscle, our glands are digesting that food for us. So those are the 2 segments of the autonomic nervous system. So this is very foundational. I want to make sure everyone feels comfortable with the breakdown of the nervous system. Again, if anyone has any questions, please let me know. Another basic information that everyone needs to know is the difference between white matter and gray matter. So the reason that white matter is white, it actually looks white, is because it has a dense collection of myelinated fibers. And the myelin, which we're going to talk about here shortly, has that white look. So the reason that it is white is because of the myelin. Okay, so white matter is myelinated fibers. That is different than gray matter. And you'll notice that I wrote gray, G-R-A-Y and G-R-E-Y. This has come up in previous classes, and honestly, both spellings are correct. They both exist. So one is not wrong and one is not right. You could really go either way. But gray matter is consistent of neuronal cell bodies and unmyelinated fibers. This is why it's gray. There is no myelin there. And when we talk about the neuron here in a second, and we talk about the cell body of the neuron, it's interesting to think that is gray matter. It's those cell bodies. And then the white matter is really those axons that are coming off of the cell body that are covered in myelin. And that's why we visually see that difference in color. So we need to know that in the spinal cord, this is a cross-section of the spinal cord, that the white matter is superficial. Towards the outside, superficial. This is all white matter here. And the gray matter is deep. The gray matter is deep in the spinal cord. Of course, nothing can be simple, right? So the brain is the opposite. Here is a section of the brain, and you can see that deep in the brain is the white matter. And superficial in the brain is the gray matter. So interestingly, when we monitor EEG, we're monitoring the gray matter because the gray matter is superficial in the brain. Now, there are a few structures in the brain that are gray matter that are deep. There are a few exceptions. Just for you to know, the hippocampus is a deep gray matter structure in the brain, and the amygdala is a deep gray matter structure in the brain. But when we generally speak about the brain, gray matter is superficial, white matter is deep, with the exception of the hippocampus and the amygdala. Versus in the spinal cord, gray matter is deep, white matter is superficial. So that was a quick review of the nervous system. Now we're going to jump into cell membrane. Again, this should be a review for most of us, but if anyone is unfamiliar with any of this information, always feel free to reach out to me. So, basic overview, cell membrane, all cells have a phospholipid bilayer. So, bi meaning two, here's one layer, here's the second layer. Phospholipids, that's what these little squiggly lines and circles are. That's what makes up that bilayer. It is asymmetric, so this looks pretty equal here, but that's not always true. This side could be thicker, this side could be thinner, vice versa. So one of the characteristics of the phospholipid bilayer is that it is asymmetric. Something important for you to know is the phospholipid bilayer has a hydrophilic polar head. So when we look at the outside of the bilayer, these circles here, we describe them as hydrophilic. What that means is that they have this polar head. So there's a charge on these. Let's just say there's a positive charge here. A negative charge, hence here. They're negatively charged. Therefore, if there is another charge that is trying to come through the cell, it could repel it, it could attract it, depending on the charge. So it's important for you to know that there is a hydrophilic portion of the phospholipid bilayer. Now, conversely, we have a hydrophobic portion as well, and that's the fatty acid tails that are shown here. These are neutral. They don't have a charge. But I like to think of the word hydrophobic. They have a phobia. So they come in together because they really don't want to be on the outside. They want the hydrophilic portion to be on the outside, the hydrophobic portion to be on the inside. So all cells have this cell membrane. Those are three characteristics of the cell membrane. It's asymmetric, hydrophilic, and hydrophobic. An application to better understand this is, how does this property of a cell, the fact that it's hydrophilic and hydrophobic, allow soap to work? So, interesting fact, cells form what's called a micelle, and this is a micelle here where all the hydrophobic tails come to the center, the hydrophilic heads go to the outside, and they form this micelle. And remember, the hydrophilic head is charged, right? So there's all these negative charges out here. And as you are washing your body, these charges are attracting other hypothetically, let's just say charges from your body, and it pulls the dirt off of you and pulls the soap, helps get the dirt off of you, and it runs down your body because the hydrophilic heads are on the outside. Now the hydrophobic tails on the inside, remember, they don't like water, they're scared, so they're protected in the inside. So that's just a fun way to try to apply hydrophilic, hydrophobic to help you remember. So another characteristic of cell membranes is that they're semi-permeable. Semi is our prefix, permeable is our base word here, and so semi meaning partial, right? Partial, partially permeable, permeable meaning what can pass through. So the membrane is permeable to certain liquids, so it allows certain things to come through. Let's just say an example, let's just say water. Water can come through. Water would be our solvent. But it's impermeable to ions, and ions have a charge, right? So remember that hydrophilic head is on the outside and it's not letting other charges get through, and that is what causes this impermeability of ions, which we're going to refer to as solutes. So I like this picture here because imagine that this door is the cell membrane, okay, and you have items on one side of the door, items on another side of the door. The door does allow the water to pass through, the solvent to pass through, but it doesn't let the solute, or all of the basketball, the wagon, the solute, it doesn't allow those things to pass through. But what can overpower the cell membrane? Just like in this picture, the osmotic pressure can. So the pressure of all of these solutes behind this door can push enough on the door to where it forces things through that the cell membrane would not normally allow to pass. So the semi-permeability, meaning what can pass through the door, what can't pass through the door, is based off of osmotic pressure. If there's enough pressure on one side of the cell membrane, that might force certain solutes to pass through that wouldn't normally be able to pass through. And we'll talk about how this relates in a second. So I do like to talk about tonicity here because it helps drive the cell membrane concepts and the flow of what goes in and out of a cell. Tonicity is specifically referring to red blood cells, is what we're going to talk about when we talk about tonicity. So many of us, maybe a long time ago, maybe recently, in high school biology, hopefully remember isotonic, hypertonic, and hypotonic. And this is based off of what comes in and out of a red blood cell. So we have our red blood cells here. Isotonic, I think of the phrase, I so happy, isotonic. It means the same amount of water that's going into the cell is coming out of the cell. Okay, so remember, the water is the solvent. Okay, we also have a solute that's inside of the cell. Let's just say it's salt, okay, for this example. So we have the same amount of salt inside, the same amount of salt outside. Therefore, we have the same amount of water going in and the same amount of water going out. So the cell is happy, iso-happy. It's isotonic. It's equal on the outside, equal on the inside. That is different than hypotonic. Okay? So in a hypotonic cell, we have more solute on the inside compared to the outside. So the analogy I like to give is, let's say you eat something that's really salty. So there's a lot of salt inside of your body. And so what happens? You get really thirsty. So you drink a lot of water to try to dilute the salt. So because there's a lot of solute inside of the cell, it makes water rush into the cell and rush into the cell. The issue with that is that the cell can get so full that it will eventually burst. Have you ever been so full of water where you just feel uncomfortable? The phrase we say to remember that is hypotonic, hypo-hippo, right? Because hippos are normally pretty large, I guess. So hypo-hippo, you're full of water, full of salt. Not ideal because eventually the cell could burst. Conversely to that, we have hypertonic. And hypertonic is going to be when there's a lot of salt outside of the cell. Therefore, the water is going to come out of the cell because the water is going to flow where there's most salt. Okay, think about the semi-permeability that we just talked about. So if the water is constantly flowing out, then the cell's going to shrink and shrivel up. So again, thinking about our analogy, I think of hypertonic, I'm really hyper. So let's say I'm running a marathon, I'm hyper. I'm constantly excreting salt out of my body. I'm sweating. So a lot of water is coming out of me and I am becoming dehydrated, right? So the cell is shrinking. So hypertonic, there's a lot of activity, you're exerting yourself a lot, you're sweating a lot, salt's coming out of your body, therefore your cells are shrinking, hypertonic. So again, to go through these, we are putting application towards the cell membrane aspect. So cell membrane is semi-permeable. It's semi-permeable based on what's inside and outside of the cell. An ideal situation is a cell that is isotonic, iso-happy. It has the same amount of water going in and the same amount of water going out because there's the same amount of solute or things inside of the cell compared to outside of the cell. So that door is there and it's happy. Nothing's pushing on it either way. Isotonic. Now, when we go to hypotonic, remember hypo-hippo. So what's happening is we have a lot of stuff inside of the cell, a lot of solute inside of the cell. And that means the water is going in the cell to try to dilute all of the things that are in the cell to the point where the cell gets so full that it eventually bursts, which is not good. Okay? And then conversely, if we have a lot of solute outside of the cell, the water wants to rush out to try to dilute all the solute outside of the cell. That's going to make the cell shrink and become hypertonic. So hyper, lots of activity, losing a lot of water, making the cell shrink. Okay? So if you're thinking, what does this have to do with intraoperative monitoring? Well, it's important to understand the basis of the movement between the cell membrane and what happens in the cell membrane. We're going to relate that to neurons and action potentials. This particular tonicity has to do with our blood cells And when you look at this graph, for those of us who like graphs, when we are at a state of isotonicity, the volume is equal to the concentration. So the cells are happy, right? We have a normal cell volume. Now, as we become hypertonic, of course, you can see here the cell is shrinking, the volume's decreasing, and therefore we're increasing the amount of millimoles, which is that solid that we have in there. The opposite way, if we have a bunch of water go into the cell, it's diluted, so we have a smaller concentration of our salt, but the cell volume is getting bigger here. And eventually we'll get to a point where the cell will burst, and that is lysis. Okay? So we don't want the cell to shrink and we don't want the cell to burst, because depending on where the cell is, if the cell begins to become hypertonic here, this is whenever we begin to have issues with our patient. So when you think about giving the patient fluid, the patient going to the restroom throughout the surgery, all of this is being done to maintain stable levels of the patient, stable osmolarity. We don't want to become hypertonic or hypotonic. We want to stay isotonic. And you can see as you become hypertonic, it can even induce a seizure. And for any of us who have been monitoring a patient and they have a seizure on the operating room table, it really goes back to their osmolarity. So I think it's important to understand not only what we're doing in the operating room, but also the roles of the other team members of the surgical team. And this is a huge part of what anesthesia does, trying to make the patient remain stable. And it can really go both ways. So when we become hypertonic, we go towards this level of dehydration. And we can have isotonic dehydration, hypertonic dehydration, hypotonic dehydration. So hyper, meaning we have excess sodium. Hypotonic dehydration, meaning we have excess water. We don't want either of those. We don't want the patient to become dehydrated. That's why we're constantly giving them fluid throughout the surgery. But then the other way, I'd like to compare hypotonicity to edema. So edema is swelling. I'm sure many of us have been exposed to swelling that happens in a patient while the duration is very long. So cerebral edema, this is whenever we have accumulation of that extracellular fluid in the brain. But what's happening on a cellular level? On a cellular level, that solvent is going into those cells, and that's what is causing the edema. Periorbital edema. This happens whenever a patient's prone on the operating room 8 to 12 hours. What is actually happening? Gravity is redistributing the fluid. It's pulling the fluid to that part of the body, which is essentially inflating those cells, and they're full of extracellular fluid. This goes back to, I like to compare it to hypotonicity versus dehydration is hypertonicity. So this is a way to take clinical correlations and relate it back to the cellular level. So that is really the cell membrane overview. I like to talk about the tonicity to better understand the flow of the cell membrane, what comes in, what goes out. Hopefully you have a better understanding of that, and then also apply it to dehydration and edema, which we can see in our patients during surgery. And that's a big role of anesthesia to try to maintain, let's say, an isotonic state. So now we're going to move on to cell membrane. So the neuron function it's highly irritable, right? Neurons respond to our stimulation. So they're highly irritable. If we stimulate a neuron, we may get an action potential. We may get a nerve impulse. But the impulse is always the same regardless of the stimulus. So if you reach that super maximal threshold, it doesn't matter if you go to 30 or if you go to 60. You're going to get the same response regardless of the stimulus. This happens because of some basic concepts of electricity that we have to understand. So, opposite charges attract, right? Negative and positive, they come together on a magnet, they stick together. Whenever we want to separate those opposite charges, we want to pull those magnets apart, we have to use energy. So we use energy to separate opposite charges across a membrane. This same concept applies to membrane. When energy is liberated, when those charges move towards one another. So let's say that we have two charges coming towards each other. There is energy that is occurring there as well. And when opposite charges are separated, the system itself has a potential energy. So when we think about the cell membrane, you think about it as two forces acting on a solute. And the solute in our previous example was salt. Okay, this is an ion, something that has a charge. So we have two forces acting on a solute in a solution. And in our previous example, our solution was water. And this is trying to cross our permeable membrane. So the membrane that's allowing things to cross. It's going to be dependent on two things. Concentration gradient, and that goes back to our picture of the door, how much stuff is on each side. The concentration on one side might drive what passes through the membrane, but it's also going to be dependent on electrical potential, and this is what we're now adding to what we've been previously talking about. If there's positive charges on one side, negative charges on the other side, those are going to be attracted to one another, and those are going to affect what move across that cell membrane. So when we think about a membrane potential, we not only need to think about how much stuff is on each side, let's say this is the membrane, concentration gradient, how much stuff is on one side compared to the other side, that's going to drive what grows across the membrane, but it's also dependent on the electrical potential. What's the charge on one side of the membrane compared to the charge on the other? And this is called the electrochemical potential. Okay, this is the potential of the membrane based on the electrical charges, that's electro, and chemical, meaning the concentration gradient. So ultimately, our cell membrane potentials are trying to reach electrochemical equilibrium. And that means they want the electrochemical potential to be zero, right? They want to be iso happy. They want for there to be a zero difference across the cell membrane. How do you think we reach zero? We reach zero by having the same amount on each side, but also having opposite charges. So if we have one, let's say for the sake of numbers, one, positive one on one side and positive one on the other side, well, that doesn't reach zero. That gives us a difference of 2. So we want to have positive 1 on one side, but the opposite charge on the other side, negative 1 on the other side, and that will give us 0. So we want the concentration gradient to be equal and the electrical gradient to have opposite charges. And that's what the cell membrane is always trying to achieve. So I want to briefly introduce Nernst's equation, and this is the equation. It's saying negative 61 millivolts over Z, and Z is the charge of your ion, by the log of the concentration inside to the concentration outside is going to give you your Nernst equilibrium. And this is an equation for charged solutes. So sodium has a Nernst equation. Potassium has a Nernst value. Chloride has a Nernst value. And that's just saying this is the value that for this ion that it is at electrochemical equilibrium. So each ion has its own Nernst value that it's always trying to achieve, and that is what drives the flow of ions across the cell membrane. So this particular equation tells you the electrical potential inside of the cell relative to the outside of the cell. Okay? You have a handout on this. You could read more about it. I don't really want us to calculate it. I just want us to understand it because we're going to talk about the Nernst value for specific ions and how that drives ions across the cell membrane. So in a biologic system, right, we're dealing with the human body. So in a biologic system, there's two factors that cause a potential to be across the cell membrane. Higher concentration of charged proteins inside of the cell relative to outside, and then ion pumps that cause a net change in the potential across the membrane. So we have specific molecules that can move across the cell membrane and can't move across the cell membrane. Polar molecules do not pass through the cell membrane. These examples are ions, so sodium, calcium, potassium, chloride. All of these cannot pass across the cell membrane. Remember, the cell membrane, that hydrophilic head has a negative charge. It's not going to let ions with a charge pass through it. It's repelling those. Okay? The only way that a polar molecule can cross the cell membrane is through some type of channel or with a carrier molecule. That's different than a nonpolar molecule. Nonpolar molecules can pass through the cell membrane. And this is lipids, steroids, oxygen, carbon dioxide, nitrogen. That's why steroids are such an effective drug, because they can go right through the cell membrane. So when you think about polar molecules compared to nonpolar molecules, think about trying to go through an airport. Okay, the airport security, TSA, is your cell membrane. If you do not have luggage, nonpolar, no luggage, you walk right through. But if you do have luggage, you're a polar molecule, you can't get through. You're going to have to go through the metal detector. You're going to have to go through one of the channels in order to get through. So that's my analogy for polar versus nonpolar. So if you are a polar molecule, you have to get through some type of channel. So there are peripheral proteins, and these are proteins that are on the outside of the cell membrane. Here's our cell membrane here. And then you have integral proteins, and these are proteins that are integrated into the cell membrane. They form pores in the cell membrane that make channels that open and close that can allow polar molecules to pass through the cell membrane. And this is important because these channels, there's different types of channels. So there's leakage channels and gated channels, and all of this is going to be leading up into an action potential. So these proteins serve as membrane ion channels. Leakage channels don't have gates. They're always open. So think about a park that has a fence around it, but it's not gated, so people can come in and out as they please. That's a leakage channel. That's different than a gated channel. Let's say a gated community, right? It's actually, the gates are closed and you have to be let in through the gate. Well, that's how these gated channels work, but what opens and closes the gate is different things. We have chemically gated channels, so they open whenever a neurotransmitter binds. We have voltage-gated channels, which are going to open based on the change in the membrane potential. We have mechanically gated channels, which open and close based on receptors that actually form in a certain way. So we're going to focus on voltage-gated channels. This is what allows us to obtain our potentials and our evoked potentials. And what allows us to get a potential or not is if we can control opening and closing that gate for those polar molecules to pass through. So when the gate is open, what this allows to happen is it allows those ions or polar molecules to rush across the cell membrane because they wouldn't normally be able to get across. Of course, it goes from higher concentration to lower concentration. So again, think if you have a bunch of items on the other side of the door and you open the door, what is it going to do? It's all where there's higher concentration, it's going to fall out to where there's lower concentration. And it's also going to go towards the opposite electric charge. So remember, it's not only how much, but it's the charge as well. So if the inside of the cell is more negative than the outside of the cell, then positive ions are going to want to go towards the negativity and go towards the negative aspect of the cell. So the charge is also going to drive what goes across the membrane. So ion flow creates an electrical current and a voltage change across the membrane. And this is the basis of what we do. This is how we obtain our waveforms. It's based on this ion flow and this electric current and this voltage change that lets us get a somatosensory evoked potential, that lets us see a motor evoked potential. This is the biologic basis of what we do. So there's 3 types of transport. Uniport is an ion going in one direction. So gray is the cell membrane. This is our channel. We have an ion pass through. Symport, you have 2 ions going through together. Sym meaning together. And then antiport, anti meaning opposite. You have 1 ion going in and 1 ion going out. We have 2 types of proton-mediated transport, facilitated and active. So facilitated does not require input energy. It cannot move against the concentration gradient. So when I think about facilitated, think about being in an inner tube going down what's called a lazy river, right? So you have the flow of the water, you're in your inner tube, you're just going down, there is no energy needed, but you cannot go the opposite way of the lazy river because you don't have any energy. If you want to go the opposite way of the lazy river, that's when you need active transport. This requires energy and you're going to go against the electrical gradient. So think about swimming in the opposite way of the lazy river. It's going to require energy. You're going against that gradient. So that's facilitated versus active. And this brings us kind of to the putting all of this information together for cell membrane, the sodium-potassium pump. So here is your sodium-potassium pump. This is a primary active transport, active meaning it does require energy. So here is your phospholipid bilayer. You have your hydrophilic head, your hydrophobic fatty acid tails. You have your protein that is an integral protein. It's integrated into the cell membrane. It has a channel, and this channel is based off of concentration as well as the electrical gradient as to what comes in and out of it. So there's a couple of things I want you to know about this pump. It is an electrogenic pump. There is a net change across the membrane of +1. So if you look here at your bottom key, okay, blue is your potassium, red is your sodium. For every 2 potassium that come in, this is the inside of the cell, 3 sodium go out of the cell. That gives us a +1 difference. We have +2 coming in, +3 going out. Okay, this ATP, that's adenosine triphosphate, it breaks down into adenosine diphosphate plus a phosphate. This is our energy source. This is giving this pump the energy it needs to get 3 sodium out and 2 potassium in. And this is critical for osmotic equilibrium. So this pump is always shooting for that electrochemical equilibrium. It wants the concentration to be equal. It wants the charges to be equal. And it's constantly working to make sure the body is in a state of electrochemical equilibrium. So it's important for you to know first, this is an integral protein. It is a gated protein. It's based off of the concentration and the electrochemical gradient. It is antiport, meaning that ions are going in the opposite direction. It requires energy, your adenosine triphosphate to your adenosine diphosphate. And this is a critical piece in our osmotic equilibrium. It also plays a critical role in creating an action potential, which we're working towards. We're working towards an action potential. So, tying this all together, my question to you is, why does more sodium go out of the cell compared to potassium going into the cell? If I tell you that sodium's Nernst value, so remember the Nernst value is where sodium wants the membrane to be at equilibrium. Sodium's Nernst value is positive 59 millivolts. And potassium's Nernst value is negative 91 millivolts. So with this information, why does more sodium exit the cell compared to potassium going into the cell? I like to bring this up because I don't like people to just memorize. 2 potassium go in, 3 sodium go out. I think if you understand why that's happening, it'll help you understand it so it's not just memorization. So I'm jumping ahead a little bit, but a resting membrane potential of a cell is negative 70 millivolts, meaning it's more negative inside compared to outside. 70 millivolts. Do you think potassium is more happy at negative 70 millivolts, or is sodium more happy at negative 70 millivolts, if this is their Nernst values? Well, potassium's more happy, right? Negative 91 millivolts is closer to resting membrane potential of negative 70. So it doesn't need to do a whole lot to get where it wants to be. But sodium, on the other hand, if we're at negative 70 millivolts, it wants to be at positive 59 millivolts. So it's not happy at all. So it is driving more sodium out of the cell to try to make the inside of the cell more positive compared to the outside of the cell, because that's its Nernst value and that's what it's achieving, versus potassium that's pretty happy with negative 70 millivolts, so it doesn't really want to go too farther than where it already is, so only 2 potassium are coming in for every 3 sodium that are going out. So the Nernst value is really the ion's driving force for its effort it's going to take to get there. So you can see from these values, sodium's going to be trying a lot harder to change this membrane potential than potassium is at resting negative 70 millivolts. So that's the cell membrane portion of the lecture. We're now going to move into the next section, which is neuron. So when we think about the nervous system, we talked about the central nervous system and the peripheral nervous system. There's really 2 principal cell types. There's neurons and neuroglial cells. So neurons are those excitable cells that transmit electrical signals. And we're going to talk about neurons here in a second. We also have neuroglial cells, also known as glial cells, and these are the supporting cells that support the neurons. And as you can see, I've listed some neuroglial cells here. They may sound familiar to you, and I've put if they are a part of the central nervous system or if they're a part of the peripheral nervous system. So astrocytes, microglia, ependymal cells, oligodendrocytes, those are all glial cells in the central nervous system. And remember, the central nervous system, it consists of what 2 structures? The brain and the spinal cord. The brain and the spinal cord. That's different than the glial cells that are in the peripheral nervous system. This is the satellite cells and the Schwann cells. And the peripheral nervous system consists of what? The peripheral nervous system consists of the spinal nerves and the cranial nerves. Okay? So these cells support the brain and the spinal cord. These cells support the spinal nerves and the cranial nerves. Okay? And all of the glial cells support the neurons that are within those parts of the nervous system. So you can go through these glial cells. I don't want to spend too much time on them. I do just want to point out that when some of these glial cells kind of go astray, that's when we get some of the clinical applications that we see in the OR. So an astrocyte, these help support the neurons. They help guide neurons to where they need to go as they're forming, as they are providing pathways in the brain. Of course, when something goes wrong with an astrocyte, you get an astrocytoma. A lot of intraoperative monitoring is involved in astrocytoma removal, so I want to make sure everyone knows where those tumors come from. You have microglial cells. Microglial cells help migrate towards injured neurons. So if a neuron is injured, these glial cells come, they phagocytize the microorganisms, meaning they eat them. They get rid of the neuronal debris and kind of clean up the area. So you can imagine if we didn't have the microglial cells, we could say all the trash would pile up. So they play a pivotal role with cleanup. Ependymal cells. The ependymal cells are going to line the cavities of the brain and the spinal cord, and they really assist in that cerebral spinal fluid flowing. I think of the ependymal cells as kind of little hairs that keep the CSF flowing. When something goes wrong with the ependymal cells, we get an ependymoma. The reason ependymomas are normally around those ventricles where the cerebral spinal fluid flows. You can see here, this is the cerebral aqueduct. We'll learn about that more later if you aren't familiar with it. This ependymoma is blocking the flow of the CSF, and that's because the ependymal cells line those structures where the CSF flows. Oligodendrocytes, this is a very important one that you need to know. Oligodendrocytes are the processes that wrap the central nervous system fibers. They form the insulating myelin sheath. So in the central nervous system, all of the white matter, remember white matter is covered in myelin, that is produced by these oligodendrocytes. The glial cells in the peripheral nervous system, we have the satellite cells and Schwann cells. For whatever reason, I always remember these were with the peripheral nervous system because they both start with an S. Has nothing to do with peripheral nervous system, but that's just how I remember them. The satellite cells surround the neuron cell bodies in the peripheral nervous system. So we'll talk about the neuron here shortly. A part of the neuron is the cell body. So these satellite cells are surrounding the cell body. And this is in the peripheral nervous system. So that is your spinal nerves and your cranial nerves. And then the Schwann cells are interesting because the Schwann cells surround your peripheral nerves and form the myelin sheath. So in the central nervous system, the myelin sheath is formed by the oligodendrocytes, but in the peripheral nervous system, the myelin sheath is formed by the Schwann cells. So always important to know structures that are the same but in different parts of the nervous system. So the Schwann cells are vital to regeneration in damaged peripheral nerve fibers. So that's also very important if you've ever done a brachial plexus or a peripheral nerve injury. They actually will essentially sew the nerve back together because peripheral nerves can regenerate, and so the regeneration is happening from these Schwann cells. Of course, when a Schwann cell kind of starts to grow and gets out of hand, we get a schwannoma. And this is an example of an extremity where we have a schwannoma. So you can see this is the nerve, and it actually grew out of the nerve. Again, that's the myelin here, and so it's an overgrowth of the Schwann cells, which form the schwannoma, and we can remove that from the nerve, and there's your beautiful schwannoma there. So those are our glial cells. Important to know those. Important to understand how their role is in the central or the peripheral nervous system, but then what's the clinical application whenever something goes awry, whether it's astrocytoma or schwannoma. So when we talk about the neuron, which is the other most abundant cell in the nervous system, interestingly, neurons are very long-lived. They can outlive the human body. We've seen that by isolating a neuron and putting it in an environment where it can thrive. It does have a high metabolic rate, so it does rely on having continuous oxygen and glucose. The membrane of the neuron allows for electrical signaling as well as cell-to-cell interaction. So this is how neurons can speak to other neurons within the nervous system. This is probably one of the more important slides for the CNM exam. We often see questions about the neuron and the different parts of the neuron on the CNM exam. So it's important to know that the neuron itself is the individual structural and functional unit of the nervous system. So when we talk about the nervous system, we're talking about the central nervous system and the peripheral nervous system, which we've already gone over. And the neuron is the individual unit of both of those, okay? When you think about a neuron, it has several different parts. It has a soma. The soma is also the cell body. So this is the part of the neuron that houses the nucleus. One important thing to know about the cell body, remember we talked about the satellite cells in the peripheral nervous system. They surround the cell body. Also, another thing that we talked about was gray matter. Gray matter is comprised of cell bodies. That's why it's gray. So it is this part of the neuron that really makes up the gray matter. That is different than the axon. So we have one axon that arises out of the cell body, and this is going to carry signals away from the cell body towards the synapse. And so when I think of axon, I think of away. Axon away, AA, carries signals away. That is the axon's role. So it carries that electrical signal away from the cell body towards the synapse. That is different than the dendrites. So the dendrites, this is one neuron here. This is another neuron here. Okay? So the axon's carrying the signal away, and then these are the dendrites of the next neuron. And they're going to take that signal and bring the signal to the cell body of the next neuron. So the dendrites transmit the signal toward the cell body. So one more time, just to reiterate, the axon carries the signal away, and the dendrite carries the signal toward the cell body. Okay? The synaptic cleft, that's really circled here in red. This is the small swelling, which is going to allow the exchange of neurotransmitters so that one neuron can talk to the other neuron. That's what's happening at that synapse and the synaptic cleft. It's the exchange of neurotransmitters, which allows one neuron to talk to another. So this is our neuron. This is the parts of the neuron. The soma, or the cell body, that contains the nucleus. The axon, that transmits the signal away. The dendrite, which transmits the signal towards the cell body. And the synapse, which is the exchange of the neurotransmitters, which allows neurons to speak to one another. Again, just going through the parts, the cell body, also known as the soma, it does consist of the nucleus. One important thing to know is it also is where the axon hillock is. The axon hillock is a part of the cell body where the axon arises. This is really where any electrical signaling originates, at the axon hillock, so that is important to know that structure. Another point to make, I'm sure we've all heard nuclei and ganglia. If you've been in a peripheral nerve case, or if you've been in a brainstem case, or even a brain case, they'll say, we're at this nuclei, or we're at this ganglia. So both of those words are actually related to cell bodies. It's just in the different parts of the nervous system. So when we talk about a nuclei, we're talking about a cluster of cell bodies in the central nervous system, versus when we talk about ganglia, that's a cluster of cell bodies in the peripheral nervous system. Dendrites and axons, these are our processes. They're bundles of processes. Whenever we use the word tracts, so we'll say motor tracts, sensory tracts. These are the dendrites and axons that are in the central nervous system. That's different than when we say nerves, okay? And nerves are in the peripheral nervous system. Again, nerves and tracts both consist of bundles of dendrites and axons, but it's going to depend on what word we use based on if we're in the central nervous system or the peripheral nervous system. Dendrites, so they are conveying the electrical signal toward the cell body. They receive input, they're short, there's normally several of them that are coming off of the cell body. That's different than the axon. The axon, there's normally one axon. It comes from the axon hillock. We just talked about that. That's a part of the cell body. The axon goes and then it branches out, like you saw in the picture earlier, into axon terminals. And this is where it secretes those neurotransmitters, which are going to talk to the next neuron. It can excite the next neuron, or it can inhibit the next neuron. We're going to talk about that as well. Ultimately, it generates and transmits the nerve impulses away from the cell body. Myelin sheath. So the axon is surrounded by myelin sheath. The function of the myelin sheath is to protect and insulate the axon. It also helps increase the speed of the nerve impulse and the transmission. I often think of electrical wires that our electricity runs through. The black covering, that's the myelin sheath. Think if you didn't have that black covering, you wouldn't have increase in the signal strength. It also wouldn't be protected if it rained. The electricity wouldn't work. So I like to think of an analogy as the myelin sheath as that black protective covering and cabling. It protects the axon, but it also increases the speed of the nerve impulse. So when we think about the neuron and we think about that myelin sheath that's around the axon, depending on if we're in the central nervous system or the peripheral nervous system, the myelin is created by different glial cells. And it's important to know that Schwann cells produce the myelin in the peripheral nervous system, and this can regenerate. That's why when we do peripheral cases and we may transect a nerve and attach it with another nerve, we're able to do that because the Schwann cells, the myelin sheath in the peripheral nervous system, can regenerate. Now, we wouldn't slice a piece of the white matter in the central nervous system. And the reason we don't do that, and when we're working in the brainstem, we have to be so meticulous, and it takes more time is because the myelin in the central nervous system is formed by oligodendrocytes, and oligodendrocytes cannot regenerate. That's why when we injure the white matter in the brain or the spinal cord, the patient has a permanent injury. That is why intraoperative monitoring is used most of the time, because we do not want to injure those structures because the oligodendrocytes cannot regenerate. So whether we're in the peripheral nervous system or the central nervous system, the myelin sheath assists with saltatory conduction. And saltatory conduction is when that action potential jumps from one node to the next. So here's the myelin in white. This is our axon in yellow. We'll have the action potential jump from here to here as it goes down the axon, making its way to the next neuron. That is called saltatory conduction. In between our myelin over our axon, we have small gaps of the axon that are exposed. And these are called nodes of Ranvier. This is also something that we've seen on the CNM exam. So nodes of Ranvier are the gaps between the myelin sheath. It occurs every 1 to 2 millimeters. And these gaps are important because this is where we have that ion exchange. So when we talked about the sodium-potassium pump, here we are. We're on the axon. In between the myelin sheath, we have this exchange of sodium. That's what excites the axon. That's what creates that signal. And that exchange of ions happens at the node of Ranvier. So, of course, as we get the signal going down the axon, a couple of words to know: propagation and conduction velocity. Propagation is the sequential generation of new action potentials down a nerve. The size and the shape of the action potential does not change. It's the variation and the frequency of the action potential that changes the information from axon to axon. So when we think of propagation, you think about Morse code. The click, the intensity of the click in Morse code is always the same. So maybe it's beep, 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 beep. But what's different about the propagation is the frequency and variation. So maybe beep, beep, beep versus beep, 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 beep means something different. That's what propagation is. The size doesn't matter. It's the variation in the frequency that communicates differently between neuron and neuron. Hope everyone appreciated that example. And then conduction velocity is the speed at which the action potential is propagating down the axon. And conduction velocity is a huge part of nerve conduction studies, right? If we have a decrease in the conduction velocity, that could suggest abnormality and it could suggest disease. We don't always look at that when we're doing intraoperative monitoring, but we do look at it whenever we're doing peripheral nerve cases for intraoperative monitoring. So it's important to know the conduction velocity. Again, we've talked about white matter and gray matter, but I'd like to revisit it now that we have gone over the neuron. So white matter is the dense collection of myelinated fibers. So again, think of your white matter as your axon. Your axons that are covered in myelin sheath, they're conducting these impulses. That is the white matter. That's different than the gray matter, which is mostly your neuron cell body. So the neuron cell body, that piece of the axon, is what makes the gray matter. So when you think about the brain and the gray matter being on the outside, the white matter being on the inside, think about those cell bodies lining the gyri and sulci of the brain, and then those axons going deep into the brain with the impulses going from neuron to neuron. Ultimately, when we talk about the neuron, there is a direct relationship between the diameter of the axon, the thickness of the myelin, the distance between the nodes, because that's where that ion exchange happens, and the conduction velocity of the nerve fiber. All of these things are related. If we have a decrease in thickness of myelin, we're going to have a decrease in conduction velocity. If we have a decrease in the nodes between the myelin, we're going to have a decrease in conduction velocity. So all of these things are directly related, and whenever we take these concepts of the neuron, if anything happens to any of those characteristics, it produces clinical correlation. Multiple sclerosis. This is an autoimmune disease where the body attacks the myelin of the central nervous system. And remember, the myelin in the central nervous system cannot regenerate. That's why this is such a debilitating disease, because once the myelin is disturbed, it can't recover. It can't come back. And therefore, you have decrease in conduction velocity, decrease in communication between neuron to neuron, and that's when you get jitteriness, loss of memory, all of these different symptoms. Guillain-Barré, this is an autoimmune disease where the myelin attacks the peripheral nervous system. So remember, our peripheral nervous system is our spinal nerves and our cranial nerves. If we're attacking the myelin of our spinal nerves and our cranial nerves, then we can't have efficient afferent sensory signals and efferent motor signals. And that's why the most common cause is neuromuscular paralysis, because you can't get those efferent motor fibers out through those cranial nerves and those spinal nerves because the myelin isn't there. The axon's not protected. You don't have the conduction velocity. So you can see how everything we're talking about is now starting to come together. And then lastly, Charcot-Marie tooth. It's a genetic mutation causing demyelination of the central nervous system. So that is, while we have demyelination of the peripheral nervous system similar to Guillain-Barré, Charcot-Marie tooth is a genetic mutation versus Guillain-Barré is an autoimmune disease. So I've attached more information about these clinical correlations. Please read more about it. These may not be patients that we're necessarily operating on surgically, but these are patients that would be related if we're doing nerve conductions, and it's also related to somatosensory evoked potentials. If anyone has any type of myelin degeneration in the central nervous system or the peripheral nervous system, you're going to see a direct correlation in latency and more than likely amplitude in any of your evoked potentials. So with that being said, this is a good place to take a break. If you need to take a break, we're going to move on to synapse and action potential, and that'll wrap up this section. So feel free to take a break now if needed.